Biology, Chapter 12, Theory Video 4, Synthesis of ATP. So now we have learned quite some details about ATP. Now we are going to discuss how ATP can be synthesized using, now we know that about phosphate groups, we know about adenosine group. So now how are we going to synthesize ATP? That is what we are going to figure out. Energy for ATP synthesis can become available in two ways. In respiration, energy released by reorganizing chemical bonds. Now we understood earlier. Now respiration means the process where energy is made available as a result of chemical reactions that take place inside the body. So these chemical reactions will actually involve the breaking down of glucose molecules or some other energy containing molecules. So the main molecule is glucose. The glucose C6H12O6 will be broken down into carbon dioxide and water while releasing a lot of energy. So this energy is what is available to produce ATP, because ATP extracts this energy and delivers it to where it is required. This is chemical potential energy. During glycolysis and Krebs cycle, right? So that energy released by reorganizing chemical bonds during glycolysis and Krebs cycle. So this chemical potential energy is made available as a result of glycolysis and Krebs cycle that we will be learning in our next stages. So that will allow some energy to come through. However, most ATP in cells is generated using electrical potential energy. This energy is from the transfer of electrons by electron carriers in mitochondria and chloroplasts. So this process is also a result of the breaking down of these food materials. It is not a different mechanism. It is once again the same process. So we will go through this and see how these electrons are involved in the energy transfer process. It is stored as a difference in proton. So proton is a hydrogen ion. Why? Hydrogen atom is a proton. It does not have neutrons. A proton and an electron. So ion means its electron has been removed. When the electron has been removed, it is simply an electron. It's simply a proton. So the proton is remaining. So H plus ion is similar to a proton. It is a proton. It is stored as a difference in proton or hydrogen ion concentration across some phospholipid membranes in mitochondria and chloroplasts, which are essentially impermeable to protons. So the phospholipid membrane is impermeable to protons. So there is a proton difference, a concentration difference between the two sides. Protons are then allowed to flow down their concentration gradient by facilitated diffusion. Facilitated means it cannot go through the permeable membrane, but there has to be some molecule to facilitate it through a protein that spans the phospholipid bilayer. So it goes through the entire bilayer. So you can remember that in AS level we discussed about phospholipid bilayer. That means the phospholipid heads and tails, etc. So that part, again, please go through that part. Then it would make it easier for you to understand how the structure is formed. Part of this protein acts as an enzyme that synthesizes ATP and is called ATP synthase. So this facilitating protein, some part of this protein is an enzyme, and that enzyme is called ATP synthase. What is its function? The synthesis of ATP. The transfer of 3 protons allows the production of 1 ATP molecule, right? Provided that ADP and an inorganic phosphate group are available inside the organelle. So don't confuse this with adenosine triphosphate part. No, it is for ADP will be converted into ATP with 3 protons. But ADP and an inorganic phosphate needs to be there. So it has to be there inside the organelle, and if it is there, these protons will be transferred and 3 protons will produce, will combine ADP and an inorganic phosphate to produce an ATP. Who does it? ATP synthase. That is what facilitates it. This process occurs in both mitochondria and chloroplasts and is summarized in figure 12.5. We will go to that and see. The process was first proposed by Peter Mitchell in 1961 and is called chemiosmosis. Chemiosmosis. So basically what happens is this. There is a concentration gradient across the phospholipid bilayer of hydrogen ions or protons. This change in proton concentrations allows the transfer of protons along the along a protein that passes through the phospholipid bilayer, and this protein also, at least some part of this proton has the ability of acting as an enzyme. This enzyme is referred to as ATP synthase. This is referred to as ATP synthase, and this ATP synthase will allow three protons to combine an ADP molecule with 3 protons and a phosphate group. It will produce ATP, and this process is known as chemiosmosis, and it was first discovered or first proposed by Peter Mitchell. So here they have given the definition of an H plus ion, that is hydrogen loses an electron to produce H plus. So now let's see how this chemiosmosis takes place. As you can see, if the diagram is not clear, please have a good look at your textbook. As you can see, there is a phospholipid bilayer. Then there is a protein channel that bypasses through both the phospholipid bilayer. So it could be the thylakoid membrane of chloroplast or inner membrane of mitochondria. Right? Then protein channel will allow 3 hydrogen ions to enter the inside of the mitochondria or the chloroplast. Right? Then you have this larger circle here. This part is the ATP synthase part, and this ATP synthase will facilitate the transfer of ADP and phosphate inorganic group to convert into ATP when 3H or 3H plus or 3 proteins come in. This ADP and phosphate will convert itself into ATP. So this is what happens. ATP synthase has 3 binding sites. as you can see here in 12.6 in this diagram. And a part of the molecule that rotates as hydrogen ions passes. This produces structural changes in the binding sites and allows them to pass sequentially through three phases: binding ADP and P1, forming tightly bound ATP, releasing ATP. Right? So those are the three steps that are involved in here. Now let's write down a note. Synthesis of ATP. Energy required for the synthesis of ATP become available in two ways. Number one, chemical potential energy. Put a dash and write. Chemical bonds being reorganized during glycolysis. Chemical bonds being reorganized during glycolysis. And Krebs cycle. Krebs cycle. Energy is released. This energy is harnessed and trapped by ATP. Harnessed and trapped by ATP. Number two, electrical potential energy. Transfer of electrons by electron carriers in mitochondria. Or chloroplasts. Makes energy available. Full stop. Continue the note. This energy is stored as a difference in proton within brackets hydrogen ion concentration across the thylakoid membrane or the inner membrane of mitochondria, open brackets and write, phospholipid bilayer, full stop. Protons are allowed to pass through by facilitated diffusion into the mitochondria or chloroplast, down the concentration gradient. Part of this protein is an enzyme which synthesizes ATP and is called ATP synthase. ATP synthase. The process of combining ADP with an inorganic phosphate group using three protons to produce a single ATP is known as chemiosmosis. Please highlight that last sentence, the definition of chemiosmosis. ATP synthase, leave a line and write, ATP synthase has three binding sites which allows the sequence of forming ATP and releasing it to happen sequentially. Then you may write down the first step, binding ADP and P1, then forming tightly bound ATP, and finally releasing ATP. If you can draw and explain this very small, you don't have to actually draw the detailed diagram of the membrane, but if you can make a sketch of this the figure 12.5 and explain or write a summary by yourself so that you have understood it clearly, it would be great. And please go through this video, and if you have understood everything properly, please do question number 12.1.